Hi everybody. Uh, let's get started. Um, any questions from last time? I jotted down a few that you guys had raised in the write-ups. So I thought I'll start with a discussion around those, and then then you guys tell me if you have any more questions. So a couple of you, at least a few of you, raised the question about where did we get this equation. Some of you had trouble with this particular term. One or two of you mentioned that this is an issue, and so on and so forth. Uh, imagine the movement of a system on a one-dimensional grid like this. So each of these circles represents a state of the system. This is our variable I, which is how many what is the number of A-type individuals that are there in the hemostat. And so the numbers below the line represent the state of the system going from 0 to n. And then we on the numbers above the expressions above the line represent the probability of reaching n when you are at that particular stage. So if you are at 0, the probability of reaching n is x0. When you are at 1, the probability of reaching n is x1. x0 is of course 0 and xn is of course 1. So we will think of our system as a one-dimensional grid like this. And the case that we are discussing, we are at state I of the system. So now, if we are at state I, the corresponding probability is equal to xi. Again, if we are at state I, the probability of reaching n is xi, and that is the probability that we are interested in. But we also know, so the left and right hand side of both these equations represent the probability that starting from here, I will reach here, right? So this obviously is the definition of that probability. So left hand side is no problem. And what I'm trying to say is that left and right are both the same probability, which is starting from here, you reach here. So left hand side is straightforward because that's the definition of xi. Right hand side is tricky in the sense that if I'm at state I, then I know if I'm a state I, then I know that in one time step, I will either be at I minus 1, either be at I plus 1, or I will remain at I. That is going to be the three fates of the system after one time step. What's important is that these three are mutually exclusive events. If you are at I minus 1, then you are not at I and not at I plus 1, and so on and so forth. And what that means is that after one, I can add the probabilities from these three mutually exclusive states. So after one time step, the 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 probability that I reach I minus 1 is pi I minus 1. And from I minus 1, so in one time step, in this particular term, in the first term on the right hand side, the scenario that I'm envisioning is that this is my current state of the system, and the scenario that I'm envisioning is that in one time step, I move to this state, and then this becomes my starting point, and I have to reach this. But that probability, when this is my starting state, the probability of reaching here to here is xi minus 1. So in case I move here in my first step, then the probability of from here reaching n is xi minus 1. But the probability of moving from I to I minus 1 is pi I minus 1. So the whole probability that I will reach n via this route, that from I go to I minus 1 and then go to n, is just pi I minus 1 times xi minus 1. Pi I minus 1 represents that from here I go to here as my first step, And then xi minus 1 represents the probability that starting from here, I reach here via whatever route possible. The second way is that I actually in the one time step, I don't change the state of the system, and I remain at I. The remaining at I happens with a probability pii, right? So in one time step, I reach pii, so I'm here. But what is the probability of reaching n when I'm starting at I? It's just xi. So the probability that I will remain here after one time step and still be able to reach n is just the probability of remaining here, which is pii, times the probability that starting from I reach n, which is xi. And similarly, the last one, that the probability of reaching n via this particular route, that from I go to I plus 1 and then reach n, is just equal to pii plus 1 times xi plus 1. Does that make sense? I mean, I think at least three of you had problem with the equation. So does that make sense?
Yes, sir.
Questions? Okay. The next question which I found was that uh, was that we are assuming that the population size remains constant at n, and there are a couple of possible critiques of this particular assumption. First is that number may not remain constant in a chemostat. The number will change as we progress through the experiment. That's one. The second is that in an ecological niche, the number is not going to remain constant, it's going to fluctuate with time. And both of these are fair critiques. Right? But remember, this derivation is obviously way oversimplified. But the point is that as long as we agree that every ecological niche is some sort of a chemostat, maybe with variable carrying capacities. So carrying capacity of an environment is number of individuals it can support. So in this chemostat, if n is the maximum number of individuals that can live, then the carrying capacity of this is n. But maybe after a while, the nutrient supply gets poorer, and hence the carrying capacity decreases from n to n by 2. So the carrying capacity is changing. Similarly, in an ecological niche, there are 24-hour cycles, so maybe the carrying capacity oscillates with time. But that's not the point. The point is that there is some carrying capacity at some point, and within that highly idealized scenario, we have found that fixation probability of a beneficial mutation is S, where S is the selection coefficient associated with that particular uh, parameter, that particular individual or beneficial mutation. Now, if we were to relax some of these assumptions and make math a whole lot more complex, and people have done that, maybe the fixation probability will change from S to 2S. So there are these many variants of the derivation that we did the other day. And what happens with each one of them is you try and relax these assumptions, and what ends up happening is that you can try, but when you reach one is that analytical solutions become untractable. So the system soon goes to a state where you have to resort to simulations and not solve it analytically, which is fine. But when you solve it, when you perform simulations on this, you find that the probability of fixation has maybe changed from 1 times s to 2 times s maximum. So, and in biology, while that's perhaps problematic in a physical sciences setting, but in biology, unless you're off by an order of magnitude, it's not that bad a result. Uh, by using that simple a logic, if you can arrive to within a factor of 2, that's actually not bad at all. Okay, so a question for you. Talking of carrying capacity, what do you think happens to these two curves? So I have a bacterial species growing in this chemostat, constant supply of nutrients, constant removal of spent waste. As this experiment goes for a long time, long here meaning several thousand generations, what do you think these graphs will look like? The first one is the initial ancestor that I started the experiment with had a growth rate R naught. As I move forward in time, what happens to R naught? And the second graph is that the initial steady state that was established, if the ancestral strain was allowed to establish a steady state in this chemostat, the number of individuals would be n naught at t equal to 0. But as mutations happen, uh, what happens to the number of cells in the chemostat? What do you think these graphs will look like? Is the question clear?
Yes, sir.
What do you think these
Yes.
graphs will look like? Anybody?
So just a guess, sigmoid?
Sigmoid meaning S-shaped.
Yeah.
Okay. Why do you say that?
Like it will reach saturation after a point, after reaching steady state.
What do you mean steady state?
Uh, the the birth and death rate maybe at equilibrium get fixed.
No, but that's not the question. The question is slightly different. The question is that once you allow this system to reach steady state, right? In these conditions, there is a certain growth rate, right? Actually, let's revisit this question a little while later. Let me just quickly go through the other doubts that are. One of the questions I just wanted to, the question was trivial, but I thought I should cover it because it's important. That why was R equal to 1 by S? R, remember, is the growth rate of the mutant. Uh, and we write it as 1 plus S because growth rates are normalized by the growth rate of the ancestor. So whatever might be the growth rate of the ancestor, 0.2 per hour, 0.8 per hour, anything, that depends on the media and the conditions in which you are doing it. Whatever is the growth rate of the ancestor, we normalize it with that. So the growth rate of the ancestor becomes 1. And then if this is a variant carrying a beneficial mutation, then its growth rate is obviously going to be greater than the growth rate of the ancestor. And since the ancestor's growth rate has been normalized to 1, we also normalize its growth rate by the same factor. And we write this as 1 plus S. And S then becomes the selection coefficient associated with the mutant that we are working with. And S can also be thought as what is the fractional improvement in the growth rate as compared to the ancestor. Feel free to stop me. Uh, then there was a comment that fixation doesn't really happen. It's a hypothetical concept. That's not true at all. Fixation is actually uh, definitely something that we see all the time. And for that, let me just these sort of studies have started coming out in the last 2-3 years a lot. This is a paper in Nature from 2019, and this is experiments done using yeast. And if you look at it, this B graph B is a fixation event happening. That you started with an ancestor, and all these are mutant lineages that came out. So this brown one really spread. It increased in frequency, right? So these are generations, and this experiment has been done for 10,000 generations. This brown lineage really spread, but then there was this red lineage, which actually shrunk quite a bit. Its frequency was really small. But then somewhere around here, I guess it picked up a mutation which was highly beneficial. And at this particular time, the fittest at this particular time when this mutation happened, perhaps the fittest lineage was the blue one because it was replacing all the other uh, alleles. But then this red one happened and it just quickly took over the population. So that's fixation happening, and there are several other examples of uh, fixation events happening. This red one here, this yellow one, blue one, and all sorts of things. So fixation happens all the time. This is not a hypothetical concept, and it happens largely in agreement with what we did. Again, if we don't take into account a factor of 2 or 3, which really doesn't make any difference. Interestingly, this paper the title reads High Resolution Lineage Tracking Reveals Traveling Wave of Adaptation in Laboratory Yeast. This is an example using yeast. Uh, so the traveling wave model is an interesting model which we are going to do next time. So if you're interested, you can read this paper after Friday's lecture. Okay, um, assumption of births equal deaths. I mean, really, this is sort of a little nitpicky because the way you should think about this is that uh, the the interval that we have chosen for the one time step, our time interval, is so small. It's not as if we fix the interval and we note the number of births and deaths taking place in the interval. It's as if that whenever a birth and a death event takes place, we close the interval. So the interval might be of a variable size, and we'll do something called a Gillespie algorithm for stochastic simulations, and this will become. But again, if there is an odd interval where two births took place or one extra death took place, it really doesn't change the result that we are worried about. There is something called Fisher's fundamental theorem, which I'm saying this in the context of, I forget the precise remark that somebody wrote, but there's something called Fisher's fundamental theorem. And Fisher's fundamental theorem, if you read books on population genetics, is covered in many contexts. But I think the one of the contexts in which we'll superficially look at it here is sort of interesting. So Fisher says that natural selection is making organisms fitter, right? So he's basically putting two and two together, just saying that natural selection is making organisms fitter, which means the organisms are getting increasingly well adapted to survive and procreate in this environment that they are in. But if that were the case, then because organisms are getting fitter, and fitness, remember, is represented by the number of progeny that you leave behind, because he's talking of sexual populations. So if fitness is getting better, natural selection in action automatically implies that fitness is increasing. But that means that the population should increase because you have individuals which are leaving behind increasing number of offsprings. So that's one. But the second observation that Fisher makes, that if you look at all the studies that have been done in the wild, populations of organisms, and remember this is like 100 years back, uh, when you look at studies in the wild, uh, if you're not interfering too much with with any ecological niche, a forest for instance, then numbers of individuals of each species, more or less, unless there was some sort of a catastrophe, either human induced or otherwise, the numbers of each species more or less remains constant. But if natural selection was indeed in action, then you should see a constant increase in fitness of individuals. So Fisher, to explain this, introduced something called Fisher's The first term on the right here is basically saying that numbers should change because of natural selection in action, because fitness is constantly increasing. That should happen. On the left, he makes the point that unless there has been a catastrophe, like a drought or something, then basically there is no net increase in numbers. Numbers of every species stays more or less the constant. So if this is happening in conjunction with natural selection in action, which would imply that organisms are getting fitter, then something else needs to happen too. And that something else, Fisher says, is the change in numbers because of change in the environment. Because if you have species, and Fisher's argument is that if you have species getting fitter and fitter, producing more offsprings, then what happens is that that leads to some sort of a change in the environment too. The environment degrades. The environment, the degradation, by degradation of the environment, Fisher means two, well, Fisher means at least two obvious things. One is that it's easier for the predators of this species to find this prey because there are more individuals now. And second is that the resource availability for this increased population is now becomes more scarce, and there is going to be increased competition for resources. So this change in numbers because of this concomitant change in environment that has taken place is actually negative because the environment has suffered uh, in a detrimental way and hence can support fewer individuals. And the balance between these two leads to what is going to be the net change in the number of individuals of a particular species. If you look at Fisher's fundamental theorem quantitatively in any population genetics textbook, what most of the books are going to focus on this particular terms, because this term describes the action of natural selection, which is the primary interest when you are studying evolution. So the left hand side and the second term on the right are more or less ignored. And Fisher then goes on to develop quantitative expressions for each one of these. And Fisher's fundamental theorem is often in textbooks only represented as this particular term, which is the action of natural selection. And Fisher shows that this rate in the rate of increase of fitness because of action of natural selection is proportional to the variance that exists in a population. If you have a population with greater genetic diversity and hence a greater phenotypic diversity, natural selection acts faster because of this greater availability of variance. If you compare to a population which is very narrow in its spread, and hence that will evolve slowly as well. So if you look at Fisher's fundamental theorem, you will see two versions. This is the complete version, and this is the first term of the theorem, which is often said to be the fundamental theorem in itself, which says that the rate of increase of fitness of a species, of a population, uh, is directly proportional to the variance that exists in that population. And I think we must keep this in mind, the complete thing in mind when we are thinking of an example such as this, that fitness cannot keep on increasing forever and ever. There are going to be constraints, resource constraints that you hit eventually. All right, questions? Okay, yeah.
Could you explain the how you can get the value of xi intuitively?
I'm sorry, I didn't hear you. Can I explain what?
Could you explain how you get the value of xi intuitively? I wasn't able to understand that.
Can I'm sorry, your voice is not very clear. Can I explain XI into PII? Is that what you said?
Uh, how you got the value of xi intuitively?
Oh, intuitively. Oh, okay. Um, Imagine this. Imagine, let's go to the marbles in a jar game. Okay. You remember that, right?
Yes.
Yeah. So let's imagine that you have 10 balls in the jar, right? Each of a different color. Okay. Now, if that is the case, let me ask you, what is the probability that the blue ball goes to fixation?
So by blue ball, do you mean probability of picking a blue ball out of the all the balls, or
No, so I'm asking, what is the probability that the blue ball of all the 10 colors, it's the blue ball which reaches fixation, and by that what I mean is that eventually all the balls in the jar are blue.
turning into blue?
Hmm?
Are the balls turning into blue color?
नहीं नहीं यार. Were you not there when we discussed marbles in a jar?
No, I was absent in that lecture.
Yeah. So you might want to take a look at it, and and then
Recording.
yeah, just take a look at the lecture, and just remind me on Friday if it's still not clear.
Yeah, actually, I saw the lecture,
Hmm.
recording of the lecture, but I
Hmm.
was still not able to understand.
So the rules of the game are that you pick up a ball from a jar. So the jar has 10 balls. You pick up one, you see that it's blue, right? And then you place this blue ball that you picked back in the jar, right? And you place an identical blue ball in the next jar. So now you have two jars. One is the original jar with all the balls in it still. And now you have another jar, jar number two, which has one blue ball, right? Then you pick up another ball from the original jar, take a look at its color, put it back, and put an identical ball. You put a ball of an identical color in the jar number two again, and you keep on doing this till the number of balls in jar number two reach 10. Okay.
Yes.
And once that happens, you ignore jar number one, and now you play the same game starting with jar number two. Okay.
Yes.
So as you transition from jar number one to jar number two, what I'm guessing you can imagine fairly straightforwardly is that some of the colors are not going to be represented, and some colors might be represented twice. You might, by that I mean, you might have two balls of the same color, and maybe there is a color of which you have three balls in the second jar. So some colors have gone extinct and some colors have increased in frequency. Does that make sense?
Yes.
So now let me ask you, what is the probability that when I'm starting with 10 different balls, I mean 10 balls of different colors, what is the probability that it's going to be blue balls? I will be left with a jar which will contain only blue balls.
Number of blue balls, number of times balls are picked.
I can't hear you, Rishi.
Number of blue balls upon the number of times the balls are picked.
No. So let me ask you this. If you're starting with balls of 10 different colors, what is the eventual fate of this game?
It's supposed to have 10 different balls. If the it's supposed to have 10 different color balls in the B jar.
Not supposed. What do you mean supposed to?
If all the balls are drawn,
No, but I want you to imagine yourself doing this exercise. What do you think is going to happen?
some of the balls will be picked two or three times, some of the balls won't be picked at all.
Correct. And if you keep doing this over and over again, remember that a color which is lost can never make an entry again, right?
Yes.
So what is going to be the long-term fate of this game?
Only one color will be remaining in the jar.
Hmm?
Only one color will be remaining in the jar.
Only one color will be remaining. So then, what is the probability that the only color remaining will be blue? What do you think?
One by number of times the balls are picked.
Number of times what?
Balls are picked.
What is that number?
And
Hmm? See, the intuition behind this is the following, that you had balls of 10 different colors, right? And you know the eventual fate is going to be that only one color remains in the jar. Now, a priori, there is no reason for you to believe that one color is going to be preferred over the others. So, any color could reach fixation, and blue could reach fixation with the same probability as green could reach fixation, right? And since eventual fate is that somebody will reach fixation, that means the probability of one individual color reaching fixation is just 1 upon n, where n is the number of colors in the jar. Does that make sense?
yes.
And if you extend that logic, you get a system, you get a solution that if there were I number of blue colored balls in the jar, then the probability that it's going to be a blue, it's going to be blue which reaches fixation is just going to be I upon n.
Yeah, got it.
Yeah, just think about it. I'm happy to come back to this again, but let me just cover a few things. So,
Yeah.
Friday is going to be something. Friday we're going to be discussing something that is perhaps going to be the most math-intensive class of the semester, and we're going to be doing this traveling wave model of bacterial adaptation. And this is work that is largely from a really nice paper that was published in 2008 from Daniel Fisher's group at Stanford. But I just want to use today to sort of build up towards that model. So imagine this. So let me two ways of representing this. Uh, I'm this is my this is time. Time could be real time or could be number of generations, and y-axis is the number of individuals. So at the start of my experiments, there were n individuals, all genetically identical, and this keeps on going till first mutant arises. And then this mutant increases in frequency, right? Which means if I were to draw a vertical line at this particular point, this is the number of mutant individuals, and this is the number of ancestor individuals at this particular time, and so on and so forth. If this, as this line is increasing, the number of ancestors is shrinking, the number of mutant is increasing. I could do this with respect to n, or I could do this with respect to the fraction of the population that is one particular genotype or the other. Right? That's a standard representation. Now let me ask you this. A deleterious mutation happens at this particular point. That means now we have one individual, so the frequency of that individual is 1 upon n. We have 1 upon n individuals which is carrying a deleterious mutation, and we have n minus 1 individuals whose frequency will correspond to n minus 1 upon n. We have this frequency of individuals which are of the ancestral genotype. What happens to the fate of this mutation?
Go extinct.
The mutation will go extinct, and what this graph will look like is that this will go back down to zero, and then we get back again our ancestral genotype again, right? In a more practical setting, like the figure that we saw in that paper, you will actually never even notice a deleterious mutation come up. Because remember, the arrival of these mutations is being found out via sequencing. And and when you're sequencing, you need each mutation to spread among the population such that it reaches some certain frequency before you are even able to detect its presence there. If it's one individual, if a mutation is present in 10 individuals in a test tube of bacteria, there is no way you can detect that mutation. So even in order to be able to even detect presence of a mutation, uh, you need it to spread among at least a few individuals, and that is not going to be the case with most deleterious mutations. And hence, in a representation like this, a deleterious mutation is actually not even observed. What if it was a neutral mutation? So now we have a variant, we have an individual carrying a mutation whose fitness is the same as that of the ancestor. What is going to be its fate?
Either of them will happen.
Either of what?
Fixation or extinction.
Which one's more likely?
Extinction.
Why is that?
Uh, it doesn't have any benefit for the population.
Right, but neither does the ancestor.
So both will remain in equal proportion, I guess.
Okay, so neither extinction nor fixation, both will coexist.
Yeah.
Others?
Coexistence.
Coexistence.
Others?
Are we considering drift here as well?
Yes.
So I think the probability to reach N is going to be much lower for that particular mutation.
Yes.
So it has a higher probability to extinction, I guess.
Yeah. Yeah. Exactly. So there is a very strong chance that this mutation is actually going to go extinct. Because remember, this is in one individual here. So the probability of reaching fixation is 1 upon n. N in the bacterial population is going to be extremely large. Hence, 1 upon n is going to be very, very small. That's in the purely neutral case. Even for it to coexist, it needs to go up in frequency because one individual, you are always, when you are one individual in a population as large as this, you are always susceptible to a random drift event. That you get lost from that chemostat and that's it, you're gone. There is nobody else to replace you. So you are extremely susceptible to drift events dictating your outcome. In order to overcome drift, you need to go up in frequency to a certain level, such that even if you were to lose 5 of the individuals carrying that particular allele, you still have sufficient remaining such that the population is not, totally devoid of. So most likely this guy will go extinct. Another way to think about this is that we did fixation probabilities, and we saw that if an individual is carrying 1% advantage, there is only 1% chance that it goes to fixation. Rest 99% of the times it gets lost because of the action of drift. So if an individual carrying 1% advantage is getting lost because of drift most of the times, then an individual carrying no advantage over the rest of the population is definitely going to go extinct much more than 99% that an individual carrying 1% advantage does. So almost always a neutral mutation will go extinct in a setup like this. In the case of a beneficial mutation, what would you say now?
So before the frequency reaches some threshold, it might go extinct
Hmm.
because probability of fixation will be less. But
Hmm.
after that,
Hmm.
it might go for fixation.
Yeah. Others? Yes, Namrata, that was excellent. That's exactly how we perceive this. That what happens is, so this is what's going to happen. Right? Two phases for a beneficial mutation. So a beneficial mutation occurs at this particular. I mean, this is a very dense figure with a lot of uh, words. Let me explain this. A mutation occurs here, and at this point, the frequency of this particular mutant is 1 upon n. It's extremely susceptible to. So let me before we do this, let me ask you this. Suppose we have a population 10 per 10. Okay. In this population, we have one individual which is carrying a 1% advantage. What is the probability that this one individual reaches fixation under all the assumptions we've made? Somebody.
What is the
The question is that we have a population of size 10 per 10, and we have one individual which carries a 1% advantage in growth rate over the rest of the population.
right, right?
Okay. So what is the fixation probability of this one individual?
Oh, 1%.
1%, right? So probability is 0.01, 0.99 probability that it will get lost because of drift. Now let me ask it another way. That I have the same population 10 per 10, but out of this 10 per 10, 0.3 into 10 per 10, which is 30% of the population, are individuals which are carrying 1% advantage over the rest of the population, right? What is the chance that this beneficial version is going to take over the population now? And we haven't done the math, but go with your intuition.
30%.
30%, did you say? Sakshi, was that 30%?
Yes.
Okay, others? I want others to chip in.
Yeah, 30% seems good.
Others? We won't move till I hear from the others. Is the question clear?
Can you repeat the question again, please?
The question is that we have been calculating, we calculated x1, which is the probability of fixation when there is one mutant individual in the population carrying a beneficial advantage. And we saw that for a large population, for a small s, that probability of fixation, which is x1, is roughly s. So if in this population of 10 per 10 individuals, I have one mutant individual, which is carrying a 1% advantage, so the population is large enough, the selection advantage is small enough, then the probability that this mutant reaches fixation is 0.01, right? The question is that now consider an alternate scenario where I have a population of size 10 per 10, but among these 10 per 10, zero point 0.3 into 10 per 10, which is 30% of the population, is this beneficial version. Okay, let's call it A. The ancestral type is B. But now instead of one individual, I have 30% of the population which is carrying this beneficial mutation. What is the probability that this beneficial fraction of the population will reach fixation? And all the assumptions remain that no new mutations, etc. Is the question clear, Andrew?
Yeah.
Yeah. Tell me.
I guess we have to use the formula that we had previously, where x of 30%, for example.
Yeah, that's the numerical value we are interested in. But we're not going to do the math. I want an answer from intuition. You're right in the sense that we've been calculating x1. In this scenario, we are interested in x 0.3 into 10 per 10, right?
Yeah.
But we're not going to do that. I want an answer intuitively. And Yash and Sakshi, 0.3 is not correct. I want you guys to think and tell me why is 0.3 not correct.
I think intuitively it will be much greater than 30%. Like
Yes,
it might be like 80 to 90, maybe even close to 1.
absolutely. Perfect. Why is that?
Like intuitively, I think there's a good part of the population which has beneficial mutation. So
Mhm.
it eventually they'll because of their faster growth rate, they'll overcome the other population.
Correct. Yes, Sakshi, why is 30% incorrect?
I guess when we derived it, we calculated x1, but in this case we are calculating x.
Let me pose the answer another way. What
Yeah.
if this mutant version was selectively neutral, that there was no change in fitness, but there was some mutation? So now you have one genotype which is 0.7 into 10 power 3, another genotype which is 0.3 into 10, sorry, not 0.7 into 10 power 10, and the other genotype which is 0.3 into 10 power 10. What is
Yeah.
the probability that the 0.3 into 10 power 10 will reach fixation? Is the question clear?
Yeah.
Yeah.
No?
So
Yeah,
would
in
it be
this case,
30% in that case?
It will
Yeah. In this case,
be, yes, that's the case. That's the case for which you've given an answer. That because it was selectively neutral, now we don't know. Anybody could go to fixation. And the chance that it's going to be one of those which is in the 30% of the population Every individual has the same chance of reaching fixation. So the neutral allele, which is 30% of the population, has a 30% of chance, 30% chance of going to fixation. That's the answer that you guys gave. Does that make sense?
Yes, so it's just like the A and B example that we saw, I guess.
Yes, but selectively neutral, right? But
Right.
now the scenario is that it's not selectively neutral, right? And and the answer that Shreya gave is perfect. That remember that when we have only a handful of an individuals in a population as large as 10 per 10, when we only have a handful of individuals which are carrying that 1% advantage, why is the fixation probability not equal to 1? Fixation probability is much less than 1 in that case, because when you are only a handful, drift can take drift can dictate outcomes. In a setting like chemostat, there is a chance that if you are two or three or one such individual which is carrying that beneficial allele, you might be washed away from the chemostat. And once you are lost from that environment, you are gone. If it was only left to selection, then there is no question about it. You are going to, because you have a 1% advantage by definition, you are going to defeat the other genotype. But it's drift. And if you're only a handful of guys in the chemostat, then there is a chance that you might get washed away. And if you're washed away, that's it, game over, right? Uh, but something changes when the population is 30%, right? Now 30% we are saying that zero point 0.3 into 10 per 10 individuals are this genotype now. That's a lot of individuals. And in a chemostat, the probability that zero point 0.3 into 10 per 10 individuals are going to be washed away is practically zero. Does that make intuitive sense to you?
Yes.
Yes.
Everybody? Is there someone to whom it did not make sense? Cool. So the argument is that when you have selection always favors the more fit genotype in this case or any other case. So selection is not a problem. Selection always is going to push for the more fit genotype to reach fixation. Its behavior remains constant. It's drift which changes. When the number of individuals carrying that beneficial allele are small, that beneficial population is much more susceptible to drift because of small numbers. When fraction of the individuals which are carrying that beneficial allele increases, now the role of drift has become irrelevant. It's largely going to be dictated by selection only. So as the number of individuals which are carrying that beneficial allele increases from one individual to zero point 0.3 into 10 per 10, the forces dictating the outcome of this particular genotype change. Early on, when the number was small, its fate was dictated by a combined effect of selection and drift. And because we saw that 99% of the times we are going to lose this individual, it means that in the early phase, when its numbers were very small, its fate was largely dictated by drift, because selection says it should reach fixation 100% times. So in that case, drift was extremely dominant. But as the frequency increases, drift becomes irrelevant, because you're never going to lose zero point 0.3 into 10 by 10 individuals from a chemostat. And now it's going to be dictated by selection only. So the fixation probability now, with all of this is assuming no new mutations occur, fixation probability now is practically 1. Does that make sense?
Yes.
All right, cool. If that is the case, this can be depicted like this. This is the point at which a beneficial mutation occurred. Then its numbers are going to fluctuate. And this is happening largely because of the action of drift. Right? Most beneficial mutations will just hover around here and then go extinct. That is why their fixation probability is much less than 1. That most beneficial mutations are going to go extinct before they are able to overcome the effects of drift. This early region where the effects of where the fate of a beneficial mutation is dictated by drift is the first phase in the life of a beneficial mutation. Remember, deleterious we're not even discussing, it's gone as soon as it comes, as soon as it's gone. The second phase is that there is some threshold. Let's imagine that there is some threshold after which, if your numbers go beyond that, now the probability that you are going to go extinct because of the action of drift are largely zero. Okay, so what I mean is that at this point, of course, this is not going to be discrete. It's not as if drift stopped acting beyond this particular region. Drift is still acting, and it's not as if it's totally drifted or totally. So the transition is gradual. But to make life easier, what we'll do is that we will define a threshold below which drift is acting and above which drift is not acting. What I mean is that if your numbers go beyond this level, the chances that you will be eliminated by the action of drift are very small, and we can define that very small to be less than 1% or less than 0.1% or anything. So that threshold will change depending on that. But then in the second phase, when drift is largely irrelevant, the fate of mutation is dictated by selection only. Right? So for any beneficial mutation, these are the two distinct phases that a mutation goes through in a population. One dictated by drift, other one dictated by selection. And if its frequency keeps on increasing, then we will reach a stage where every individual now is carrying this beneficial mutation. And then we say that the mutation is fixed in the population. The threshold below which a mutation's fate is thought to be decided by drift is when its fraction in the population, I'm sorry, this is not fraction, this is number of individuals. So in a large population, when a beneficial mutation of selection coefficient S, so S, remember in our example, S is 0.01. When number of individuals carrying that beneficial mutation exceeds 1 by S, so in this case 100, when number of individuals carrying that beneficial allele exceeds 100, then it's safe to assume that now it's very hard for the population to be lost because of drift. And hence it will reach fixation in this particular context. And that makes sense because the larger the beneficial, the larger the S, the greater the benefit that this mutant enjoys over the ancestral population, the faster it's going to spread, right? And the faster it's going to spread, the faster it becomes independent of drift. So if drift has to act to remove a mutant which has a very large S value, it has to act very, very fast. Because once, if a S is very large, if there is a mutant which has extremely high S, then even if it becomes 10, that could be very large. Drift might not be able to get rid of it because those guys are multiplying so much faster than everybody else. So the susceptibility to drift is hence a function of what is the selection coefficient associated with the mutation. Does all of this make sense?
Yes, sir.
Yes.
All right.
Have they done experiments to prove this, that it's of the order 1 by S, for example?
Yes. Yes.
Okay.
So now if we have this, we have two scenarios in which evolution can take place. Scenario one is that I start with an isogenic. Anirudh, to answer your question, many examples. I mean, this is not even up for a contest now. This is not even news anymore. So
Okay.
in scenario one, you start with an isogenic population. There is a time for which you have to wait, and then a mutation occurs. Remember, when we're talking mutation now, we're only talking beneficial mutations. We're not even discussing neutral mutations. a mutation occurs, and this mutation is now susceptible to drift, and eventually it crosses a threshold, after which it's largely dictated by selection, and it reaches fixation at this point. Then there is no other mutation in the culture, in the chemostat, and you have to wait a certain amount of time for another beneficial mutation to arise. And again, that mutation is susceptible to drift, but eventually it crosses the threshold, and then it's largely independent of drift and reaches fixation. There are two time scales associated with this particular behavior. The first time scale is called tau wait, which is the time I have to wait between two beneficial mutations to occur in the chemostat. Remember, beneficial mutations are occurring in between here also, but there are many beneficial mutations. In fact, most beneficial mutations are going to get lost because of drift. So the tau wait that I'm talking about is the time I have to wait between two beneficial mutations that escape drift. I'm not talking about the beneficial mutations that do not escape drift and are hence lost in this intervening tau wait time. So that's the first time scale. The second time scale is that once this beneficial mutation escapes drift, what is the time it takes to reach fixation? Let's call that time tau fix. Now, the characteristic of this particular scenario in a chemostat is that if I were to take, if I happen to be passing by at the chemostat at time t2, if I take a snapshot of every individual, right, and I get genome of every individual sequenced, then the population is isogenic. If we did it here, anywhere, we will only see one particular genotype in the chemostat. On the other hand, if you took a snapshot here, you would see some individuals of this type and some individuals of this type. But the point is that in this particular regime, no matter when you stop by to take the snapshot, you will never see more than two genotypes coexisting in the chemostat. For this to happen, we need to have this scenario that tau wait must be much larger than tau fix. Right? This time must be much larger than tau fix. Remember, this is all based on average time. Tau wait is obviously going to be a variable between mutations, but it's going to have certain distributions depending on the parameters, basically n and mutation rate associated with the species. So this is one particular regime in which evolution could take place. The second regime that could take place is that a mutation happens, but before it reached fixation, in fact, it was only here, another beneficial mutation took place which escaped drift. Then it was going up, but then another one came up and another one came, and so on and so forth. So while maybe this one reaches fixation here, then you have this as the tau fixation, but tau wait is actually much smaller than tau fix. And the qualitative difference between this and that is that now if I were to take a snapshot of this system at any particular point in time, I am going to see several genotypes coexist between several genomes coexist in the chemostat. And the characteristic of this particular regime is tau wait is much smaller than tau fix. Do these two scenarios make sense?
So can you explain the second one?
Yeah. So in the second one, imagine this, that in the second scenario, what's happening is that a mutation happened here, and this mutation overcame drift and started increasing in frequency. But before it could reach fixation, another beneficial mutation came up, and that started increasing in frequency. Before either one of the two could come even close to fixation, another beneficial mutation took place, and so on and so forth. So the time you have to wait for a beneficial mutation to come up is actually much smaller than the time it takes for a beneficial mutation to reach fixation. So tau fix in this case is much greater than tau w, tau weight, right? As a result of that, because fixation time is much greater than the time it takes for a new mutation to arise and escape drift, what that implies is that there are going to be several beneficial mutations which are coexisting with each other in the population. If I take a snapshot of this particular chemostat at this particular time, I'm going to see this genotype. This remember is the ancestral genotype, and this is one genotype, another genotype, another one, another one, another one. Right? So I'll see 6 different genotypes coexist in the population. Whereas in this case, for large parts, if this condition is met, for large parts, I will only see one particular genotype, but I may see a maximum of 2 uh, in this particular region. Does that make sense, Sakshi?
Yes.
All right. So,
Sir,
yes.
we are still talking about like all these mutations are still corresponding to one particular locus, right?
Not anymore. It could happen anywhere. But
Okay.
go ahead, make your point.
No, I was just thinking because then more than two mutations can simultaneously exist, right? If it's not corresponding to one particular locus anymore. Or is it always that one mutation occurs, then that should get fixed, and then the next mutation should occur?
I'm not sure I understand the question. Can you say that again?
Mm, no, okay, so I'll think over it. I was just thinking if this is happening in one particular locus or in all other locus because, yeah, that's fine, sir. I just wanted to know that. I'll get back to
No.
the field.
It's not fine. See, if this is my genotype, right, and I'm growing
Mm,
population in a certain environment, right? Let's say an
yeah.
advantage. Let's say a mutation takes place here, and this
Huh.
mutation confers a 1% advantage to the population, right?
Huh.
And now we have competition between the ancestral genotype, which is going to lose, and this particular mutant, which is carrying that 1% advantage, right?
Hmm.
Now, the dynamics of this are going to play out, in a weird way. That suppose suppose there is another mutation here now, which confers
Hmm.
a 2% advantage, right?
Hmm. Hmm.
Now, which one of the and remember this hasn't happened yet. We are still waiting for this mutation to happen.
Hmm.
Now, this mutation has happened, however, 1% advantage, right?
Yeah.
To begin
Hmm.
with, the number of individuals which are carrying this particular mutation is small, and the
Hmm.
ancestral genotype is much larger. Agreed?
Yeah.
Now, because the number of individuals with the ancestral genotype is very large, then
Hmm.
there is a greater chance that one of those individuals are going to pick up this mutation,
The second one.
the second one,
Yeah. Yeah. Huh.
right? Because it's just a matter of chance,
Yeah.
right? And because you have more
Yeah. Yeah.
individuals, more chance, right?
Yeah.
So
Yeah.
what that means is that there is a good chance that this new mutation, 2% mutation, is going to
Huh.
occur in a individual which is actually the ancestral genotype.
No.
Huh?
Can you repeat that? The
Yeah.
last thing that is.
No, I'm saying that there is a greater chance of the 2% mutation being picked up in an individual which is the ancestral genotype, not an
But
individual
then,
which is this particular genotype.
because the first mutation happened and it has 1% advantage over the rest of the population, that's
Mhm.
increasing in number, right?
It is increasing in numbers, yes,
Huh,
but but
so
but we don't know yet how long is tfix,
huh. Okay. Huh.
right? If
Huh.
tfix is like if tfix is really fast, then you're right, but we don't
Yeah.
know that yet. We
Yeah,
don't know
yeah,
which
yeah.
of the two regimes, right? So
Yeah.
So now let's imagine that we have a few individuals carrying this like 10% of the population carrying this, right?
Okay.
We have 90%, 89% of the population carrying this. But now we have 89.9% of the population carrying, I'm sorry, 89.9% of the population, the ancestral genotype, and 0.1%, which arose from the ancestral genotype carrying this particular mutation,
Okay.
right? So now we have ancestor versus 1% mutant versus 2% mutant,
Yeah. Yeah.
right?
Hmm.
And the chances of 2% mutation occurring in the ancestral genotype is greater if tau
Yeah.
fix is not very small, is not very fast,
Yeah.
right? So what happens in cases like this is a term called clonal interference, and I'm going to come to that in just a second.
Okay.
Okay, let me just go forward a bit. Maybe we'll come back to this in 10 minutes time.
Yeah. Yeah.
Okay? But fair point. Okay, so we want to get an estimate of tau weight and tau fix and see which one is more relevant representative of what happens in a bacterial culture going evolution in a chemostat. Is it progressing like this or this? So for that purpose, we want to estimate tau weight and tau fix. Remember, tau weight is the waiting time we can measure in generations before one beneficial mutation escapes drift. If population size is n and mutation rate is mu b, right? So we typically report mutation rate as any mutation, but in this case, we are only interested in beneficial mutations, hence mutation rate is only mu b. Then number of beneficial mutations in one generation is n mu b. Any questions with this? That should be straightforward, right?
Yes.
Now, if number of but this gives me number of all beneficial mutations occurring in one generation. That's n mu b. But I know that not all survive drift, right? So I know that each one of these beneficial mutations survive drift with a probability s. With probability 1 minus s, these mutations are lost because of drift, right? So of all the beneficial mutations that are occurring in one generation, the ones that survive drift is actually only equal to n mu b times s. Does that make sense? Yes, no, something.
Yes.
Yes.
Okay. Of course, I have assumed here that all beneficial mutations have the same S, and we'll come back to that on Friday. Right? But let's assume that all beneficial mutations have the same S. What that means is that if in one generation this many mutations survive drift, then just simple unitary method to say that how much time do I have to wait for one beneficial mutation to occur and survive drift. Again, this is how many beneficial mutations survive drift in one generation, which is this many. So applying unitary method to find that how much time do I have to wait for one such beneficial mutation to arrive and survive drift, and that's just going to be 1 upon n mu b s. So this straight away gives me tau weight. Does everybody get that?
Yes.
Okay.
Yes.
All right.
Yes, sir.
Now the second one is a little tricky, tau fix, because tau fix is actually made up of two different components. One is time time a beneficial mutation takes to overcome drift, right? Tau d, and the other one is the selection dictated phase. Let's call that tau s. So tau fixation, right from the point of occurrence to reaching fixation, is actually equal to tau d plus tau s. But remember that we are only interested in beneficial mutations that have already survived drift. So we actually don't care for the drift phase because we are only talking of beneficial mutations that have already survived this drift phase. So tau fix largely is what we are saying tau s. I'm saying largely because even here drift is not totally irrelevant. It might be totally irrelevant here, but it's not irrelevant here. So we say that tau fix is largely equal to tau s. Once it crosses this threshold, it's largely independent of drift. So at the end of drift phase, number of mutants is 1 by s because that's the definition that once you cross this threshold, you're independent of drift. And I know, and this growth is largely exponential. So I say that starting from here, when the number of mutant individuals is 1 upon s. I'm sorry, I mixed these up when I was making these notes. This 1 by s is obviously numbers, and this is obviously fraction of the population. But we can convert this to fraction. So I mixed the two representations, the number representation and the fraction representation. So if I'm going by number, this exponential growth can be modeled as number of mutant cells at time t is simply equal to n naught, which is at the starting point. N naught is just 1 by s because at the start of the period which I'm considering, the number of individuals carrying this beneficial mutation is 1 by s. So n naught is 1 by s. And exponential growth is represented by e to the power st, where s is the advantage that it grows, s is the advantage that it enjoys over the ancestral population. And I'm interested in the time that this takes for the mutant version to reach this n. And this is simply, I plug n here instead of n naught, I plug s and e to the power st, and then I take this s to the other side and take log of both sides, so I get st equal to log of ns, and t therefore, which is the time of fixation, I get as log of ns divided by s. Does this make sense? Tau fix as this. Questions with this? Okay. Now, if that's fine, then what we are interested in, that what sort of experimental conditions would ensure regime where tau weight is much greater than tau fix. Remember, tau weight greater than tau fix is this particular regime. When would I get to see this particular regime? So I want tau weight to be much greater than tau fix, and I equate the two s's on the denominator get cancelled, and the condition that I need for this to be true is that 1 upon n mu b should be much greater than ln of ns. And basically this is decided by the error rate, and selection coefficient is something that's environment dependent. So these are not experimental handles that I can play with. Of course, I can create strains of bacteria which have different mutation rate and hence different beneficial mutation rate, but largely I don't have much control over mu b. I have even less control over s. So the only variable that I have control over is n. So I'm interested in how should I design this experiment such that this condition holds. And for this, mutation rate of E. coli is 10 power minus of the order of 10 power minus 3 per cell per generation. So beneficial mutations, if I take it to be 10% of all mutations that are occurring are beneficial, we can change that number. It doesn't really matter. If 10%, I mean, it doesn't really matter in the sense that I can change 10% to 2% and I will get the same result. If this is 10 power minus 4 per cell per generation, and the selection coefficient of each of those beneficial mutations is 0.01, I am interested in values of n where this condition holds. Now, before I show you the results of that, I want you to tell me that suppose you are designing a evolution experiment with bacteria. What is the typical population size that you'll be working with?
10 power 9 or 10 power 11.
10 power 9, 10 power 10. That sounds very reasonable, right? So what I have done here is just plotted the two against each other. And remember that so x-axis is the value of n. And remember, for us to be in the first regime, we want 1 upon n mu b to be much greater than ln of ns. And in this graph, we want basically the blue curve to be much greater than the orange curve. I made some of these 0 because when taking log, it goes to negative values. So the more, and log of a negative is not defined, so I just plugged it to be equal to 1. But this curve actually goes down like this, right? We want the blue curve to be much greater than orange curve to be able to say that we are in the regime where you have one mutation occurring, reaching fixation, and then waiting a long time, another mutation occurring, reaching fixation, and so on and so forth. And as you can see that these two cross each other, and this is log scale and this is population size, right? These two curves cross each other at about 1,000, right? This is 100. So the much greater than inequality that we hoped to ensure actually works when the population size is 100. And we can't do experiments with population size of 100 simply because when I'll be working with a culture, I wouldn't even know that there is bacteria in that culture, and drift would be dictating everything that happens in that culture. So, which leads me to this result now, that now what is happening? Excuse me. Regimes of rare mutation is very hard to study because I need to design an experiment with a population size of 100 to be able to study rare mutations. And if you think about it, if you have 100 individuals, now not only do you have to keep those 100 individuals at that population size, but the time you have to wait for a mutation to occur is going to be inordinately late because mutations are so rare. So first thing is that studying this regime is really, really hard. The second implication of this is that multiple mutations coexist in a culture. If we go back to this graph, for the most part, ln of ns is much greater than 1 upon nu, which means that tau fixation is actually much longer as compared to tau weight. And as a result of that, multiple mutations coexist in a culture. Before one mutation is able to reach fixation, another one happens. And this phenomena is called clonal interference. Clonal interference is basically the phenomena where beneficial mutations are competing against each other. If there are two beneficial mutations, so Prachita, to answer your question, now if you have one type of beneficial mutations at this particular locus, but then one of the ancestral cells picks up another beneficial mutation at another locus, now these two beneficial mutations are going to compete with each other, and only one of them can get fixed. So this sort of working in a regime like this seems to be as if, so all these are beneficial mutations which are competing against each other, and only one of them can win. All the other beneficial mutations are going to get lost. So clonal interference has this property that not only are beneficial mutations getting lost because of drift, which is what we studied, but beneficial mutations are also going to get lost because of competition among different beneficial mutations. And hence, the chance of reaching fixation for a beneficial mutation is actually further reduced than what we studied. Because what we studied was when there was no other beneficial mutation, and drift and selection were the only two forces between these two genotypes only. But now we know that two genotypes only is an extremely hard condition that's probably not met in very many places. So that regime is sort of an unrealistic representation of what happens. So it seems like clonal interference further reduces the speed of evolution, and by speed of evolution, I mean the rate at which your fitness is increasing. On the other hand, supply of beneficial mutations in this particular regime is much greater. In this particular regime, you have to wait a long time before another mutation can take place. In this regime, beneficial mutations are competing against each other, but the supply of them arriving into the chemostat is actually much faster. So these are the two forces that are occurring in this particular regime. Next, I don't want to make, I only have 2 minutes. I can't mention this now. So let's stop here. Next, what we will do is, now in the scenario where we know that clonal interference exists, we have competition taking place among different beneficial mutations in a culture, and that is going to change the dynamics of evolution from what we've been considering so far. So one of the popular ways of studying clonal interference dictating evolution is this model called traveling wave model of evolution, which was first proposed in 2008. We'll do that on Friday, and I also want to take a little bit of time on Friday to discuss the questions that I will be giving you guys for the mid-semester exam. Right. So let's stop for today, unless there are one or two questions. I can take one or two quick questions and then we can leave. Tell me. Okay, if there are no more questions, I will see you guys on Friday.
So,
Yes.
sir, are the mutations that we are comparing related to like one aspect of the genome?
Well, that's a tricky question. Usually, what happens is that if you're evolving E. coli or any other organism in a particular environment, then selection pressure, let me put it this way, that when you're putting pressure on when you're growing E. coli in a certain environment, the number of ways it can increase fitness is very, very many. Okay, so suppose you are growing it in environment where carbon is limiting, where there is very little carbon. Now obviously any mutation that helps it make its carbon utilization better is a beneficial mutation, right? But the number of ways it can do that is many. So in that context, you have many beneficial mutations which are available, right? Does that make sense?
Yes, sir.
And another thing, I don't know if you guys have done a course in biochemical pathways or something, but this is how a biochemical pathway is, that you have enzyme 1, substrate converted to product 1 by E1, product 1 converted to product 2 by E2, and product 2 converted to final product P by E3. So you have E1, E2, E3 acting in conjunction. How many of you do not know what is a control coefficient? All right. Okay. Fine. So I'll describe accordingly. Now, how many of you are familiar with the concept of rate limiting step? Cool. Everybody. Excellent. Perfect. So now you have 3 enzymes, right? And let's imagine that the selection pressure is that this pathway needs to be made faster for the bacterium to grow faster, and everything else doesn't really matter. Again, a highly idealized scenario, but you have to make this pathway faster. The common fallacy in our understanding of metabolic pathways is that there is one rate limiting enzyme here, that there is an E2, and if you make E2, which is the slowest step, if you make E2 better, you need to make E2 better for this pathway to proceed further. That's incorrect. Uh, there might be an enzyme, E2 in this case, which if you make better, you confer a larger benefit to the mutant. But even if you make E1 and E3, and E2 can be as slow as you like. Even if you make E1 and E3 better, the pathway will still go faster, albeit by small amounts, but it will still go faster, right? So there is the concept that there is a rate limiting step which you absolutely need to work at in order to make a pathway better is actually a fallacy. And this is mathematically captured by something called a control coefficient. Control coefficient is basically that if you make some change here, what is the output change, the ratio of these change? That some measure of by how much do you change the flux here, and by how much does the flux in the overall pathway change. I'm making things oversimplified here. But what's very easy to show is that the control coefficient is actually spread unequally, not equally, among the three enzymes in this particular pathway. If this is the slowest enzyme, then this will have the most impact on changing. This will have the most impact on the total flux that's flowing. But E1 and E1 and E3 are not irrelevant in the context of us making a mutation and changing the overall flux. They also control the flux that's happening. So there's nothing like a rate limiting step as such. It's just that when we say rate limiting, we are we are we are assuming, we are making an assumption that we are going to ignore E1 and E3 and whatever change they make, uh, and we are going to only focus on the enzyme which makes the maximum change in the flux flowing through a pathway. Now, in the context of our discussion, what that means is that you make a change in E1 and you confer a small benefit to the advantage. You make a change in E3, you confer another small benefit to the individual. You make a change in E2, you make a relatively larger benefit to the individual that's carrying this particular mutation. So within this pathway, within studying, within the limited prism of studying a simple biochemical pathways, we have different beneficial mutations competing against each other. Right? Now you can imagine a population where mutations are being picked up in these three different enzymes, and these beneficial mutations are competing against each other. Each of these enzymes in turn, one individual could pick up one mutation in E2 and make it better. Another individual could pick up another mutation in E2 and make it better, right? So in that context, you can have beneficial mutations in one enzyme itself, different mutations being carried by different individuals which are competing against each other. You could have mutations in a pathway which are competing against each other. You could have different pathways which are competing against each other. So there's actually no limit to the number of beneficial mutations which could be competing against each other. Does that make sense at all?
Yes, of course. It's a point
Yeah.
of disadvantage.
Yeah, I mean some of the environments could be more targeted. Suppose you are evolving an environment where there is a sub MIC level of antibiotic present, right? Then it gives the cell a huge advantage if you were to pick up a mutation which helps you completely resist that antibiotic. And that if that can happen via change of the antibiotic target, that's great, right? So then you are going to select for very directed type of an evolution. But if the selection pressure in which you are evolving the cells are of a very generic nature, then the number of mutations that could confer advantage is actually very, very large. All right, I have another meeting, so I have to, Akshat, quickly.
Sorry, accidental.
Accidental. Okay, cool. Sure. So I have another meeting. We can start with a discussion on this or anything else you like on Friday, and then we'll take it.
Thank you, sir.
Bye, everybody.
Thank you, sir.